Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For the section to discuss NCRT Class 12, the chapter Biotechnology and its applications. Part 1, Biotechnology. As you would have learned from the previous chapter, essential deals with as an industrial scales production of bioinformatics and biological using genetically modified microbes, fungi, plants and animals. So the previous chapter, that is principles of inheritance and in this biotechnology principles and process, we clearly learn about as any that enter the production of the in a bioinformatics and biological genetically modified microbes, fungi, plants, and animals we learn. Am I right? Yeah. Let's. These applications, the biotechnology include as in a therapeutics, diagnostics, genetically modified crops for agriculture, production, the processed food and bioformat, the bioremediation and waste treatment and energy production, three critical research or the biotechnology or. So in this total biotechnology applications, we clearly to learn about as any there under the therapeutics there in this biotechnology including as what the therapeutic to diagnostics, genetically modified crops in agriculture, processed food and bioremediation, waste treatment and energy production. These are okay. Here, what are the main three criteria as in this biotechnology? See the first one, the providing the best catalyst in the from to improved organisms, usually microbes or pure enzymes. So the first one is we can use as the best their catalyst to improve that in a organisms using as in a what microbes or pure enzymes. And second one, creating optimum condition through as an engineering for catalyst to act that. So to create as an optimal conditions for through engineering for as an catalytic to act. And third one, downstream process. Downstream process technologies to purify any protein and organic compounds. It is to get completely downstream process the purify as a protein or any organic compounds. Let's now learn how do the human beings have used biotechnology to improve the quantity of human life, especially to feed food production and health. So nowadays, the how do the humans are using as in this biotechnology applications for a human life process as well as in a food production and health also. Let's see discuss one by one. First one, the biotechnological applications used in agriculture. First, biotechnology applications used in agriculture. The first one, let us to look at as any optional that can be the thought for increasing the food production. The first purpose we are using as a production of food. The first one, they are agrochemical based agriculture, organic agriculture, and third one is genetically engineering crop based agriculture. So the food production we are using as a three ways. The first one is agrochemical based agriculture, means we are using as a total in a agrochemicals, means like using as in pesticides. Mostly we are using as in a pesticides. Okay, and second one is what? Organic agriculture. Here organic agriculture, it is in a little bit amount only we are developed as in a their crops. And the third one is genetically modified or genetic engineered crop based on agriculture. Here mostly we are using this one only. Okay, let's see here. The Green Revolution, the success during the tripling, the food, the supply, but it was not encouraged to feed to grow a human population. So here, Green Revolution. The Green Revolution is developed by the scientist who was normally poor lad, right? His success was in tripling the food the supply, but it was not enough to feed to growing human population, because yeah, the human population will be continuously growing, the food production will be there decreasing. So that purpose we required as a more production, right? Let us increase increased yields having a they are partly being due to as in that use improved crop varieties, but mainly due to the use as in a better management practices and use as in agrochemicals, fertilizers and pesticides. So here, when we are increasing as any the crop yielding, the better management practices when we are using as any pesticides and they are enter the pesticides and fertilizers, then here what happened? It is most expensive. Let's see here. However, the farmers to developing world, the agrochemicals often too expensive. So that entire the fertilizers as well as any their pesticides most expensive, and further increase yield with their extensive varieties, it is not possible using as any the conventional breeding. So if in case when we are using that one, what happened that next academic year it's not comes as in the proper completely the yieldings are not yet. Okay, next. Here, as traditional breeding technique failed, the key place with as in a demand and provide the scientifically fast the efficient system for crop improvement. So for crop improvement process, we are using as the number of demand is present for the traditional breeding process. Here, the another technology we are using as that is tissue culture. So here to development of the total crops, we are using as any one completely the traditional breeding process we are using that to improve as a little short duration period we developed as an excess amount of the yielding. That process we called as a tissue culture got developed. What does tissue culture means? What is a tissue culture? It means that scientists that during the 1950 that is all plant would be generated from X plant. So any part of that plant we get uptake, we grow to develop as an entire whole plant. That is called as any what? Tissue culture. Any part of plant taken out and grow test tube under sterile conditions in the special nutrient medium. So it is this completely growing as in where in a particular nutrient medium. Okay, and this capacity to generate whole plant from any cell explant is called as totipotency. So this entire the pot to get generated as in the total plant to generate as in a total plant, that is called as in a totipotency. That is called as what? Totipotency. Next. Here you will learn how to accomplish these higher classes. How to they are accomplished. Let's see here. It is important the stress here that nutrient medium must provide carbon source such as any sucrose and also inorganic salts, vitamins, amino acids, growth regulators like auxin, cytokinins, etc. So if in case you get grow as any plant artificially, to grow as any plant artificially to get completely we require as what? To completely to require as in some mediums. We require some mediums. What is that? They completely we require as in that sucrose, so inorganic salts, vitamins, amino acids, plant growth regulators like auxins and cytokinins. These are completely required. Then only this plant takes place as they grow. Okay. By applications of these methods, possible to achieve to propagation of a large number of plants with the short durations. So when you are using this all simply plant growth regulators as well as in minerals are all completely sterile conditions we are applied to as in this plant within a short duration period to get developed to as in the entire plants, means number of plants also we get develops in the short duration period. Okay, next, this method of the producing thousands of plants through as a tissue culture is called as any micro propagation. So here at a small, it is a less time of period, we developed as a number of plantlets. So this process is called as a micro propagation process. Each of these plants will be genetically identical and original plants form which as they were grown. That is, they are in isoma clones. Simply, they are genetically identical. What you developed as in this all plantlets, their genetically is what? They are identical. So this all original plants, they are completely growing as an original plants, am I right? So this entire we call as what? Soma clones. We are called as in a soma clones. Next, after Many important food plants like tomato, banana, apple, etc. So these all plants we developed from this process only. Example they given tomato, banana, and then apple. Next, have been produced on commercial scales using the method. Try to visit in a tissue culture laboratory with your teacher to better understanding and appreciate as in the process. So when let's say as in to go to as in that any that any laboratory, culture laboratory, and you once you see and observe these entire things for better observation. Let us. Another important application of the method, it is a recovery of a healthy plant from diseased plant. If in case any plant they get disease, the disease to automatically they are transferred to another plants. If they are plant, they are infected with as in a virus, that is meristem, the apical or axillary, they are free of virus. So which that completely which part they get cause disease, they can automatically get removed. That is what it is a part of as in a completely they are free of virus. Hence one can be removed the meristems grow in vitro to obtain virus-free plant. So when you can remove that growing plant, we call as in what? We are obtain virus-free plant. The scientists have the success in this culture meristem, banana, sugar cane, potato, etc. So the number of scientists also get as a success for this process. Okay. Next, the scientists have even the isolated single cell plants and after their digesting of the cell walls have been able to isolate a naked protoplast surrounding by the plasma membrane. So here the number of scientists also they are digesting as a cell wall, they are isolated, they are naked protoplasm. Here isolated protoplasm from the two different varieties of plants, each having as a desirable characters. So when you notice as a two different kinds of the protoplasm, they show as a they are complete they differ as in a one by one. Here can be fused to get hybridized their hybrid protoplast, which there can be their further growth for that new plant. So the hybrid protoplast here, hybrid protoplast, it is again to develop as a new individual plant. That hybrids are called as the somatic hybrids. So here completely the hybrid protoplast also to get developed individually, that is called as what in a somatic hybrids. While the process is called as in a somatic hybridization. The process is called as what? That is in a somatic hybridization. Next, here imagine the situation when the protoplast of tomato is fused with as in a potato. So here tomato and potato, the protoplast get fused, then what happen? And they grow, they're grown to form as a new hybrid plants coming, they're combinating their tomato and potato, the characteristic. So here we getting as any one the tomato and here one potato in this both combination to develop it as in what the new crop will be there getting new characteristics will be there developed. Well, this has been achieved resulting the formation of tomato. So we getting as what this in a, that is tomato. Actually it is a tomato, it is in a potato. So finally we get result as what? Tomato. Means that unfortunately this plant did not have any that all desirable combination characters for any the commercial utilization. Okay. Remaining we discuss in next class. Thank you for watching. Please share and like. Thank you.